Hi, and welcome to the next webinar in our new Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you're experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspp.org. If you're having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all of the associated materials within a few days. If you have questions during today's webinar, please let us know using the GoToWebinar chat box. Today's talks will each be 20 minutes, followed by 10 minutes of questions. Jürgen Klein-Venn, who is an associate professor at the University of Natural Resources and Life Sciences in Vienna, is with us today to moderate your questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. This webinar is the third in our new virtual research talk series, which was initiated thanks to the efforts of Benjamin Schlesinger and Jürgen Klein-Venn as a response to the closure of most universities due to the COVID-19 pandemic. Please visit our webpage for more information on this series and sign up if you'd like to be considered for a speaker. We have had a lot of people sign up as volunteers, so it may be a while before we can get you on the schedule. You can find us on social media at plantae_org and at ASPB. For those of you listening to this as a feel free to reach out to us on Twitter with follow-up questions and comments using the hashtag PlantaePresents. Thanks again for joining us today. I'm now going to turn it over to Jürgen to introduce our speakers and moderate today's session.
Thanks a lot, Katie. Yeah, I'm very excited that we are back here on air, and we are welcoming again a big crowd of plant scientists that are very eager to watch the talks of Detlef Weigel and Remco Stam. And as Katie said, so this COVID-19 outbreak kind of accelerated the development of such virtual events as we are holding right now. It's anyway easy to envision that such virtual seminars from now onwards will be part of our way how we communicate science. And I personally thank ASPB and especially Katie Rogers and Mary Williams for putting plant science here at the forefront of this, which is really great. And well, if you like what you see, consider supporting us. This Global Plantae Presents seminar series is for free, but of course it comes with some costs. So if you want to donate something, feel free to use some of these buttons that you see on the webpage. So today I'm particularly happy that we have Detlef Weigel. He joins us with a nice green screen that he just told us that he freshly purchased for this event. Detlef got into plant science as a postdoc with Elliot Meyerowitz, so he had a very successful PhD and then decided to turn into plant science. And he establishes his independent research as assistant and later as associate professor at the Salk Institute. Already in 2001, he moved actually back to Tübingen because that's where he also did his PhD. At that time, he got a Max Planck directorship. And if you're not familiar with the German system, so in principle, a Max Planck director, this is like there's a scientific heaven on Earth. Detlef is a highly decorated plant scientist, a lot of awards, and again, I just referred to Wikipedia. There's a very nice article on him, and you can check all his many awards. Detlef is changing his research interests from time to time, so it's ranged from flower development, microRNA, genomics, evolution, plant pathogen interactions, and along these fields, he left a big trail of excellent papers behind, and not only papers but also provided a lot of resources for the plant science field. And as a side note, in later times, he in Germany he gets actively involved also with politicians, and he intends to change here the public opinion about genome editing. And today, Detlef will talk about epistasis, the spice of life, and evolution lessons from the plant immune system. Detlef, once again, I thank you for accepting our invitation, and please take the stage.
Thank you, thank you. So is my PowerPoint showing? Katie?
It sure is.
Yeah, okay, good,
Mhm.
perfect. All right, okay, good. Well, Jürgen, thank you very much. Katie, thank you very much. I've been a member of ASPD for almost 30 years. So I think this is another really great initiative. This is a very special time for all of us. I think this is wonderful that we are now moving online and having these sort of interactions. And I have to admit, I'm as nervous as I would be at a regular seminar. So I'd like to tell you a little bit about the work that we've been doing on the plant immune system. Before we start out, I briefly want to acknowledge the key contributors. I'm going to mention a number of people as we go along. Still in the lab are Christina Barragan and Lei Li, and then having moved on recently, On-Young Che, who is now an assistant professor at National University of Singapore, and Dan Koenig, who is at UC Riverside. So I'm yes.
Not to interrupt, it looks like your screen is actually blank right now, so we're just seeing a white screen.
You are seeing.
We're not in slides yet.
So okay, so it said it. It says it's showing the. It says it's showing.
We saw a graph there for a
second. Let me see.
We
Stop showing screen. And now show screen. Does this work? What do you see now? So it said
this works. Yes. Now we see it.
Ah, okay. So here it said it was showing it. Okay. So I briefly go back. Sorry about this. All right. Okay. Sorry about the hiccup. So, all right. So I acknowledge Christina and Lei, who are still in the lab. On-Young and Dan have relatively recently moved on. On-Young to National University of Singapore and Dan to UC Riverside. So I'm a geneticist by training, and genetics is what I love. I've even written a little essay on this, published in the journal Genetics. And one of the reasons I love genetics is because of various genetic phenomena, and epistasis is one such phenomenon that I find enormously interesting. Without epistasis, genetics would be very boring, because if you cross two genotypes, A and B, with certain phenotypes, the hybrid AB would be strictly intermediate. Fortunately, there is evolutionary magic, which is epistasis, where you cross two such genotypes and then the hybrid have a phenotype that exceeds that of the parents. And of course, you all know maize. It's the basis of much of modern maize breeding and cultivation. But there's also a dark side to epistasis. Epistasis as evolutionary disaster, which is hybrid weakness or negative heterosis, where the hybrid is much inferior to either parent. So how did we get into hybrid weakness? So Jürgen already alluded to this, that I've been changing fields from time to time, and we got into this really by accident. So Janne Lempe, about 15 years ago, she was interested in natural variation in flowering time, and she had these two strains of Arabidopsis from this village of Umkirch near Freiburg, and one was flowering late, the other one was flowering early, and she thought, well, they come from the same place, maybe it's just a single mutation that makes them different in flowering, so why don't I just cross them to each other? And that's what she did. But a few weeks later, she came to me and said, well, these plants actually never started to flower. And instead, this hybrid looked really poor and didn't quite grow. So at the time, we were developmental biologists. We didn't quite know what to make from this. We stared at these plants. We looked at them under the scanning electron microscope, but wasn't clear to us what was going on, but we thought this is really quite interesting that we had this very different phenotype in the hybrid. And then a little bit later, Kirsten Bomley's joined us. She's now a professor at ETH, and Kirsten did a number of more crosses between wild accessions of Arabidopsis thayana, and she found several more cases. So we actually had something to study. And since we couldn't figure out what was going on, we decided to ask the plants what was wrong with them. And the way we did this was just to look at their gene expression patterns. And so we had these three different hybrids, and you can see they look actually quite different morphologically. This one really, really small. This one is not so small, but the gene expression patterns were all very similar. So similar sets of genes induced, similar sets of genes repressed in these hybrids. And the only gene ontology categories that were overrepresented among these genes related to immune response. And this is work that we did together with the Dengue lab. And so what we figured was going on that these plants were suffering from autoimmunity and that a hyperactive immune system made them really small. So a little excursion here. They are dangerous microbes everywhere. Plants know they are dangerous microbes everywhere, and this is why they have an immune system to protect themselves in case of attack. But these plants, apparently, they just think they are microbes, even if they are no microbes. So these are sort of paranoid plants. So our hypothesis was that what's going on here is two genomes mix, and one genome contributes something that recognizes something that comes from the other genome as foreign or pathogen derived. And this phenomenon, we now know it's called hybrid necrosis. We were not the first ones to actually discover this. It turned out when we went back to the literature that hundreds of cases had been described by breeders and naturalists, though not in maize, curiously enough. And this just shows two cases from wheat. So this was really a larger phenomenon which we thought was worth studying. And when you read the literature, it was clear that the underlying defect apparently was autoimmunity, although none of the papers straight out said this. So we went on to look at the genetics, and we found that the genetics of hybrid necrosis in Arabidopsis thayana is simple, just as it is in these other species, at least where it had looked at. So this QTL map told us there were two genes, which we call dangerous mix, DM, DM1 and 2, and this is just a different way to look at these QTLs and their interaction, producing in epistatic manner this phenotype. The first gene we cloned was DM1, and we were super excited because it mapped to an NLR gene. So NLR stands for nucleotide binding site, loose and rich repeat receptors. These are very important immune proteins. You'll hear more about these from Remco later on. Suffice it to say that these proteins, this shows a very nice cryo-EM structure from Gigi Chai and Zhang Zhu, published last year. These proteins directly or indirectly can detect the presence of foreign proteins, of pathogen proteins. And they are known to be among the most variable genes in plant genomes. This is what you can see here. They are up to 20% amino acid differences between the different alleles. So this is the reference alleles. There are two copies of this gene. Then the strain with the good allele has also two copies, but neither of them is apparently functional. And then this strain here, which has the bad allele, has only one copy. And as I said, 20% amino acid differences, which is huge, but not unusual for these genes. So we were very happy. And then we expected that the other gene, DM2, would just encode some random plant protein. We were in for a surprise because it turned out that DM2 also encodes an NLR immune receptor. And we then later on found out that DM1 and DM2 actually heterodimerize. And down here are shown other alleles of DM2 that give rise to other types of incompatibility, and I'll show you that in a second. So what do we know about how NLR proteins function? This is one way of thinking about this from this review by Franz Tacken and Tamerling, that these proteins basically are enzymes that cycle back and forth between an off state and an on state, and normally you need a pathogen protein, an effector, to get them from the off to the on state. And apparently this DM1, DM2 heterodimer is just too often in the on state, and therefore it signals even though there is no effector present. So around that time then, Eun-Young Choi also joined us, and Eun-Young said, let's look at this in a more systematic fashion. So how common are these type of epistatic interactions really? So we became Arabidopsis breeder, essentially. So at the time, 2010, we had sequenced the genomes or resequenced the genomes of 80 strains of Arabidopsis thaliana coming from 8 different geographic regions. And Eun-Young essentially crossed everyone to everybody else as male and female, something that a breeder calls a complete diallele, an almost complete diallele. And you can see there are these incompatibilities scattered all across this interaction matrix here. And they are a different severity of hybrid necrosis. So the worst ones are these purple, well, these pink ones here. And then a lot of them are relatively weak. You can barely see them in the F1, and they only show up really strong when you go to the F2. So what we've been thinking for a while, that we're sort of looking at the tip of the iceberg. So these are something like 150 cases in these 6,000 crosses. So tip of the iceberg in the sense that perhaps there are more cases which you don't see morphologically, but where there is a sublethal activation of the immune system. So at the time, these strains had been sequenced. This is why we use them. So mapping genes had become a lot easier. And here you see the 5 chromosomes of Arabidopsis thaliana. And then in color are shown where you have NLR genes colored by density. And you see that almost all of the causal genes for this hybrid incompatibility, this hybrid necrosis, map to NLR immune receptor loci. And even the ones which do not map to NLR genes, they map to genes that somehow are implicated in immunity, with very few exceptions. And you have cases like this at DM2, what I introduced before, where one locus produces several risk alleles, each of which interacts with locus somewhere else in the genome. And this is really was from the beginning my favorite case, where we have 2 loci, RPP7, which encodes an NLR, and then this RPW8HR, which encodes another type of protein. And we have 3 alleles here and 3 alleles here that produce hybrid necrosis. And so this is something that Christina looked in more detail, and it's really a specific interaction. So the RPW8HR allele at Merck0 gives hybrid necrosis with the RPP7 allele of KZ10, but not with phase 0 and DON0. And LARIC1-3 only interacts with phase 0, but not with the other 2 accessions here. And then Eun-Young, together with Christina, looked at the genomic region where you find RPW8HR, and they found that this region sort of looks like what we see for an NLR cluster. So this is not a member of the NLR family, but you again have this crazy diversity, so a lot of copy number diversity. So look at this expansion here, and also a lot of sequence diversity. And was very fortunate because Lei Li joined the lab, coming from John Minju's lab. He was an expert in biochemistry, something that we weren't very good at. And then Lei was actually able to show that the biochemical mechanism for this interaction was not only direct interaction between RPP7 and the phase 0 allele of HR4 here, but that HR4 phase 0 acted apparently as a ligand that induced oligomerization of this RPP7B NLR from that is associated with the hybrid incompatibility, and that you see here on this Blue Native page, that only if you have the 2 bad proteins together that this large molecular weight complex forms, but not if you have the bad RPP7 with a control HR4 or the bad HR4 with a control RPP7. You see very little of this, if any. So how do we think about this? Without going into details, we think that these RPW8 and HR4 proteins, they probably make pores in the membrane, and that helps the plant to defend itself against pathogens. And what we think is going on, that normally a pathogen will inject an effector, its own protein, into the cell that somehow will modify this protein so it can't do its job anymore. And then the RPP7 receptor detects this modified HR4 protein and initiates signaling. And what's going on with this HR4 phase 0 case is that this protein, without being modified by the effector, already looks to RPP7B like a modified version, and therefore you get signaling in the absence of a pathogen. So from all of this taken together, we think there are many potentially promiscuous interactions between NLRs, and these likely limit the possible immune receptor combinations you can have in a single genotype. So the way we think about this, having one resistance, again, one pathogen is good. Having two resistances against two pathogens is even better, and so on and so forth. And so you ask yourself, why is it that we can't find a single strain that has all the resistances that are available within a species? And the answer is apparently, or one answer is, if you have too many of these resistances, then bad things happen because these proteins and their clients start to interact with each other in a manner that is bad for the plant. And this conclusion is supported by findings from other, for example, this paper from Frank Tucken, this paper from Sophie and Kamoun, that there is a large network of NLR interactions. Okay, so this just summarizes what I told you here. So these epistatic interactions are common in the immune system. Many translate into direct protein-protein interactions. We think too much diversity at NLR and other immune loci can backfire, and study of the natural autoimmunity has really revealed new aspects of the plant immune system. Now, you'll hear more from this, about this from Remco, that NLR immune receptor genes are very polymorphic. And this is from a study that we published last year together with the Dengel and Jones lab. And basically what this just showed, when you look at NLR genes, and we did this in 64 different accessions of Arabidopsis thayana, what you see is that most of them are restricted to very few accessions, and there are very few that you see in most or all of accessions. So there is this amazing diversity of NLRs. And of course, one of the big questions, unsolved questions, is why are NLRs so diverse? And the first answer we came across a little bit by chance. This was work by Dan Koenig, and he had been studying these two species of Kapsella rubella and Kapsella grandiflora. And Kapsella rubella was formed only something like 30 to 200,000 years ago, when probably a single individual of Kapsella grandiflora became selfing. And you see that these all form a nice clade that clusters within the glandiflora, larger diversity of the species, and there's very little diversity compared to what you have within grandiflora. And so because this species comes from within this species, not very surprising that there are many trans-specific alleles. But then Dan later also looked at another species, Kapsella orientalis, which split about 2 million years from these other two species. And much to his surprise, he also found many trans-specific alleles shared between orientalis and these two other species. So why am I telling you this? Well, I'm telling you this because not only is there a very large number of trans-specific alleles in these species, that these trans-specific alleles are much more common than you would expect. So orientalis suffered a recent very strong bottleneck. So in 16 individuals, only something like 70,000 SNPs. But a very large fraction of these is actually shared with grandiflora and rubella. And where are these SNPs? Well, these SNPs are these trans-specific SNPs. Almost all of them are at NLRs and at other immune genes, and the same for rubella and grandiflora. That there is an enrichment for sharing of alleles between species at these immune loci. So what we conclude from this, you can lose diversity in much of the genome, but you cannot lose diversity at NLR genes. So this then, so we see that there is diversity, and this diversity must somehow be maintained. So the question then is, how does this actually work? in the real world. So we understand the concept, but what is really going on in the real world? And why is it that often the same loci and often even the same alleles where you have a retention of diversity? So orientalis, for example, you have here in Central Asia, whereas rubella and grandiflora are around the Mediterranean, but you maintain diversity at the same loci and the same alleles, although the pathogen environment here is probably very different from the pathogen environment here. So what my lab has now pivoted to is really trying to understand what drives immune system diversification in the wild. And this really, I think, nicely parallels what you'll hear about from Remco in a couple of minutes. And so we started 2 years ago this project, Parsodopsis, Pathogens on Arabidopsis, where we have gone across all of Europe to collect Arabidopsis and its pathogen. And this is a project that's driven by Rebecca Schwab and Gautam Shirzekar. And this is really what I want to leave you with, this wonderful quote from Rosemary and Peter Grant from their book, How and Why Species Multiply. Nothing in evolutionary biology makes sense except in the light of ecology. From being just geneticists, we became evolutionary geneticists. And so we now realize that in order to understand this evolutionary diversity, we have to understand the ecology of the plant as well. And so this is what I want to all leave you with. This is what it looks like when you prospect for Arabidopsis in natural places. All right, so this is the end of my presentation. And now I turn it back to you, Jürgen, or?
Yes, perfect.
All right. Okay, good. All right, good.
Thank you, Detlef, for this wonderful talk. I will select some questions here from the audience. The first one from Jeff Dean. I think it's a particularly interesting one. He is wondering whether anyone found examples where this mechanism, so he's pointing at the hybrid necrosis and its link to autoimmunity, may have provided a pathway to speciation events.
Yeah, so it's a bit our fault that we put this in the world, if you will. So when we first discovered this, we were really excited about this because hybrid necrosis actually is mentioned as an example of Dobzhansky-Müller incompatibility, and this is also why we call these DM genes, and it's mentioned as such an example in the book by Coyne and Orr on speciation. So we thought that this would be a way to study potential speciation mechanisms. I would not exclude that this could lead to speciation. It is really difficult to know how often this will lead to speciation. So for Arabidopsis thayana, these hybrids they do form in nature, but they are relatively rare. So it's unclear how important it is as a selective force to avoid hybrid necrosis. So we really now think about this more, that the main evolutionary mechanism is avoiding having too many of these genes in the same genotype. But it is indeed formally, it is one of these mechanisms that could lead to speciation.
Mm-hmm. Freddy Monteiro is wondering if you ever tested if the basal expression level of the incompatible NLRs, either on the parental lines or F1, has any effects on the severity of phenotypes observed in the F1.
Yes, so that was, of course, something that we considered in the beginning, that it could be either at the expression level or it could be at the protein-protein interaction level. So in the cases where we looked, it was always protein-protein interaction. So we have gone back in some of these cases where we know the causal allele, and then we have asked by sequence which other strains have the same causal allele at the protein level. And when we tested it, we always saw the interaction. So that suggests that expression differences are not so important, that if you have this, then you'll get the incompatibility. What we have seen in terms of expression, Christina is right now investigating one incompatibility where there seems to be a runaway effect, where we have interaction between two NLRs, but then we have interaction between two NLRs, and then we have massive induction of almost all NLR genes in the genome in this hybrid.
Mm-hmm. So another question from Taras Pasternak is he's wondering about the, I mean, maybe a bit a similar way, like about the cell type and stage specificity of such a finding.
So we haven't looked at whether this is restricted to just a part of the plant. So this phenotype, this cell there seems to affect much of the plant. So we haven't really, asked, are there differences between root and leaf? And so that you have this very strong phenotype apparently comes from this process happening in many different cells. What we do know is that this is dependent on temperature. So it's known that activity of the immune system is very much temperature dependent, and that actually helped us with genetics. So almost all of these cases, when we grow them at high temperatures like 23 degrees or even 27 degrees, we can suppress the phenotype. But we have actually one case that even growing the plants at 27 degrees, they still die.
Mm-hmm. And Indu Sharma wonders whether there's some benefits of epistasis to have more resistant plants against pathogens.
Yes, so that certainly could be the case. So there's really lovely work on interaction between NLRs in crops, and you have both situations where you have different NLRs that suppress each other, or NLRs that help each other. So we haven't formally demonstrated this, but it's certainly possible. And so, for example, our hybrids, if you grow them at a temperature where they don't die yet, as you would expect, because many immune genes are switched on, they are more resistant to certain pathogens.
Mm-hmm. Well, coming to another level of regulation, Ido Keren wonders whether epigenetic effects modulate the autoimmunity.
Yeah, so again, when we started this, we had no preconception whether it was, transcription regulation, whether it was epigenetic relation, whether it was protein-protein interaction. So somewhat unfortunately, we only found protein-protein interaction, but I hope that, somebody will find other types of interactions. It certainly would make perfect sense. So we know that some NLR genes are strongly regulated by epigenetic phenomena. So it would make perfect sense if one would find cases where epigenetics plays a role.
Mm-hmm. So there are a couple of questions, and they are wondering whether there are differences between Arabidopsis and for example, maize immunity, and whether this can also explain difference in hybrid necrosis.
Yeah, so this is something that is really odd, and Ed Buckler has told me that he has looked really hard for hybrid necrosis. He says, he has looked at hundreds of thousands of plants and never seen anything like this. And none of my maize friends have come up with a good explanation. Of course, I haven't come up with a good explanation either. But what is also curious, that compared to other crops, most of the resistant genes that have been cloned because they provide resistance to agriculturally important pathogens do not encode NLR genes. So there are NLR genes in maize, and a couple of them correspond to classical resistance loci. But from what has been cloned, it seems that this is not the main type of resistance that maize relies on.
Mm-hmm. So there's a question from Naoki Yanagisawa, and he wonders about the comparison between plant autoimmune and animal autoimmune system, and whether there are similarities, and whether plants would also produce some toxic chemicals in response.
I'm convinced that not just animals, but even humans. So I'm convinced that there are cases of human infertility that have to do with sort of hybrid incompatibility of the immune system. And there is indirect evidence from human genetics that there are certain alleles at different immune loci that do not seem to be compatible. Somehow it's something that, there is not a lot of literature. So you can't set up crosses with humans as you can with plants and do these systematic crosses. But I bet that does exist. I always wanted to do this with Drosophila, but never got around to it. So.
Mm-hmm. And maybe a last question, again, an interesting one from Ho-Han Go. And he's wondering about gene silencing and why it doesn't impact on LRR genes to maintain the hybrid vigor.
Yeah, no, so we know that NLRs are very tightly controlled. So there are all these different layers. There's transcriptional control. There's post-transcriptional control through microRNAs, which target many different NLRs. And so absolutely. So we thought about this, that hybrid vigor could be sort of the opposite of hybrid necrosis, that you could dampen immune activity, and that would give you more vigorous plants. So again, I would love to find this, and we have looked hard for this. It has just turned out to be technically really challenging, but if somebody would be able to demonstrate this, that would be really, really cool. And I wouldn't be surprised if that is a mechanism that could contribute to anti-hybrid necrosis as a part of heterosis. Totally.
All right. Thanks a lot, Detlef, for the nice talk and also for the great discussion. And
all right. Thank you, Jürgen.
Well, I could only pick some of the questions that were posed here. And if you still have questions, or if you are watching the recording of this and you still have some questions, please turn to Twitter. So Detlef is very active at Twitter. Use the hashtag Plantae Presents and maybe add Detlef's Twitter handle at Plant Evolution. And I think like this, we can all join the discussion then over there. All right, thanks once again. And next up is Remco Stam. Remco initially joined the group of Ralf Hückelhoven at the TU Munich as an Alexander von Humboldt Fellow. And it is typically you keep the good ones. And so he became subsequently a junior group leader there at the TU Munich. And his main research project focuses on wild tomato species and its pathogens. And Detlef already put some references to his upcoming talk. And we are very happy to have Remco and to have him sharing his creative science. And he will talk about the diversity and molecular evolution of plant defense against pathogens in nature. So please, Remco, it's all yours.
Thank you, Jürgen. Can you hear me all right? Okay, so, what happened there? Can you still see my screen? Because I can't.
Yep, we can see your screen.
Oh. Okay, well, anyway, so then, yes, so let me make one correction. So I started as a Humboldt Fellow in the lab of Weigel Tellier, where I really learned doing population genetics, and now I moved on as a group leader in the department of Ralf Hückelhoven, but that doesn't change the topic. So today I want to talk about what the factors are involved in resistance evolution in nature. And I think everybody knows about all these things. There are many factors out there, right? So, and Detlef hinted on a lot of them already. We have pattern recognition receptors, R genes, physical barriers, metabolites, etc. And since you need to start somewhere, and Detlef did such a good job introducing one of the main factors in resistance, we also started looking at R proteins in specific. But my lab now is also moving on, and I would also like to show you a little bit about the immune response, so actually the readout of the defense later on, if I have enough time for that. So, unlike many other people, I will need to introduce my model system. So we are working with Solanum chilense. I started this work, like I said, with Weigel and Tellier. And at that time, people had just realized this is a wild tomato species, and it has basically 4 genetic groups that you can define. You can define a group in the north, you can define a group in the central region on the border between Chile and Peru, and 2 groups in the south, one in the highlands and one on the coast. And in between here, there is the Atacama Desert. So actually the southern highland and the southern coastal are geographically the furthest apart, right? This is what these sites look like. So in the highlands, you have some places where the plants really can hide from the sunshine, where there is humidity. On the coast, it's extremely barren. There's not so much going on. And if you're in the central range, You go from the coast all the way up to the mountains, up to 3,500 meters altitude. And especially in the higher ranges, there is a lot of humidity part of the year. So there's, we think, a large variation of pathogen pressure. And this is why we think that within this one species, we can very well study evolution of resistances and differences in resistances, because we expect very different pathogens to be present, or very different pathogen pressures to exist in these different locations. And that is actually what we found. So we did a very simple assay. We took plants from a seed bank and we infected them with different pathogen species. And to make a long story short, this has been published a few years ago now, we can basically show that wherever the population comes from, the north, the south, the center, there are always some plants that are very resistant, be it to Alternaria, Fusarium, or Phytophthora, and there are always some plants that are much more susceptible. So we really have a system here that has some variation in its resistance. We also have recently published our own reference genome, and the thing that I would like to focus on particularly is that we put a lot of effort with the follow-up study in mind to annotate the NLR genes. So we find 236 NLR genes. We double-checked them. We did a lot of additional analysis. We backed them up with expression data and whatnot. And the most important findings that we have are that everything looks to be relatively complete. The number doesn't really differ that much from other wild tomato species. We find, very importantly, this NRC clade here, which is kind of a helper clade, so it needs to dimerize with the guys on this side in the graph for those to function, and we find all the others that can work on their own or homodimerize. So we have a very complete set of NLRs, so we have almost all the resources that you need to study our gene evolution in a nice system. The last thing that is lacking is the demographic history, and we also know this for Solanum chilense. Of course, we could have guessed from the map with the central region, the area of origin, and then the two branches in the south being the more derived populations. By using whole genome resequence data for 3 complete plants, we could estimate indeed that this is the case, but what we found to our surprise is that the split was not at the same time, but the population on the coast split a lot longer ago, 200,000 years ago, than the population on the south. So there's a lot more admixture, a lot more interaction between the south and the central region, the mountain south. And this is very interesting because this gives us now multiple evolved populations that we can start comparing with each other. So then we set out to sequence our genes. Now we are not Max Planck. I'm not a Max Planck director at all. So we don't have enough money to sequence full genomes, long reads, etc. So we decided for a pooled resequencing strategy, 14 populations, 10 plants per population. By enriching and sequencing in pools, we could very cost effectively get a lot of information from these plants. We do have the problem that since we didn't use long reads, we needed to map to a reference, which meant that we lost a lot of data during quality control and filtering. So the methods for this have been published in 2016 in Genome Biology and Evolution, and what I now show has been published last year. So the data that we have are in the end from a set of about 200 down to 100 NLRs, where we can show that we have very robust data on SNPs and evolutionary parameters. So if you study evolution, you can calculate a lot of statistics, because that's what we wanted to do. We didn't want to look at presence, absence of these genes. We wanted to know how a gene, when it's there and when it stays, how it evolves over time. So not that it disappears, like that I showed in some of his graphs, but which nucleotides are changing and which one is changing when it goes to which new area and whatnot. So all these statistics, the only two that you need to keep in mind are nucleotide diversity in general. So the higher the number, the more diverse, which is an indication of general diversity and how large your population is, and whether these polymorphisms are synonymous or not. When they're non-synonymous, that means that the amino acids change, which means that the R gene or the R gene makes a different R protein, which likely has an evolutionary effect. Later on, I'll also talk a bit about fixation index. I'll come to that later. Okay, so what do we have? 14 populations, 10 plants per population, 200 plants each, what we can call the SNPs, and this is where we are. I forgot to say, we have a set of control genes in our data set, just to know what the baseline is in this species. And here we can already see that what we expected holds true. So we see some variation in the center, right? And we see lower variation in the south and the coast. Makes sense. These are the derived populations. And we see higher diversity when we move further north. Right. Then we look at the NLI genes. We see actually that the medians and actually the whole box plots, they really mirror our control genes. So on average, these NLRs in sequence diversity terms are really not that much more diverse. But what is very important here is this cloud at the top. So there are definitely a lot of outliers there, a lot of NLR genes that have higher diversity than our control set. Now, is this diversity synonymous or non-synonymous? Because non-synonymous is probably more important in evolutionary terms. Well, yes, it is. So here we see our control genes again. All the values are well below 1, which means equal ratio. Here in our NLR genes, we see that the values on average are still much below 1, but the cloud here is already a bit higher. So we definitely see that there are these differences. So NLRs have a higher nucleotide diversity and also more non-synonymous mutations. But then you can start looking at this data in many different ways. So we can split, this is the same data, but now plotted for the different clades that we have. And this is becoming interesting. So CNL21 and CNL20 are new clades. We found them for the first time in Solanum chilensis. They don't exist in Solanum pennellii or in Solanum lycopersicum. So this is cool. They're more diverse, new clades, makes sense. And on the other side of the spectrum, we see this NRC clade, these helpers, and they are very conserved. And this is also what has been hypothesized, that these helpers, they need to collaborate with the sensors, so they need to maintain some kind of conservedness to actually be able to allow this dimerization. This was work that Sophie Kamoun presented and worked on. So this is cool, but what we really wanted to see is now which R genes are evolving, how many are really evolving throughout the species, and where do they go. So now I've plotted all the R genes and our control genes actually, by N by S, for each individual gene where we could get reliable data. And now you see that actually, yeah, what they actually see. I mean, there's no clear outliers in terms of non-synonymous diversity. There's some here that are definitely higher up. Interestingly, none of them could be assigned to any of these known clades. But that's it, right? How do you make sense of this? We see the color coding here is actually interesting. We see that in some cases, the high diversity is, in this case, for example, in the south, and here it's in the center, and here it's on the coast. So still doesn't really make much sense. And this is where the demography comes in. So we have one other statistic that we can calculate with the data that we have, and that's the fixation index. So this fixation index basically compares the diversity within and between populations. And the only thing that you should remember is it goes from 0 to 1. And when it's at 1, it's high. And when it's high, that means we have different alleles, and the gene probably changed the evolutionary pressure. So it means it's under balancing or diversifying selection. So there we go. We plot all the FSDs for all the possible pairwise comparisons that we can make. So we compare population gene 1 in population 1 with population 2, gene 1 in population 1 with population 3, etc., etc. 91 populations, 91 comparisons. It's a lot of dots. And again, we see a nice cloud, we see a nice distribution, but there's not really much that we can read into this. And moreover, we know that if species or populations are further apart, you expect higher diversity. And if we plot this, so I did this here, I colored, for example, the south and the coast, or the coast and the north, I plotted in this bright yellow or dark blue. You can see that they're always at the top, and that makes sense because they're the furthest apart from each other. So the further apart, the more diverse. So with this plot and just these FSD values, you still cannot really say, well, this gene is constantly evolving because there might be a new pathogen somewhere in the south of the species range. So there we go. Now we need to start using our demographic model. This is work done by Gustavo in Professor Tellier's lab at the TU as well. So when we build a model using an ABC model where we basically assume the current situation and also how these populations were connected back in time to simulate the diversity that you would expect under normal circumstances. I mean, these populations are evolving, they're migrating, they split either 50,000 or 200,000 years ago, so you will expect some diversity. The question is, are our genes more diverse than what you would expect in a neutral scenario? And that is what we actually see, and we see it very clearly. So we simulate for 30,000 genes in each possible pairwise comparison again. So here I've simulated one population 4330 and the other population 4118. We simulate FSC for NLR genes, but then we simulated 30,000 times, as if the whole genome was NLR genes. And then our simulations say the absolute top value that you can expect is here, 0.6. What we then find when we look at the real data or calculated FSC is that there are here 1, 2, 3, 4, 5, maybe 6 NLRs that are outside this range. So these have an extremely high FSC for this comparison. Then we compare this in all the other possible scenarios. I plotted only 4 more here, but we did 91. And you see that in almost all the cases, there are some NLRs that are outside this simulated range, well outside the hypothetically expected distribution. And these are the genes that we think are under very strong evolutionary pressure. They're diversifying or they're under balancing selection to maintain this diversity in the species. So in total, we find 53 outlier genes, not so much. Out of about 100 where we could reliably assess the data. So we find some genes, here's one number, just an example, where we don't find anything. But for the others, we can plot how often we see the gene coming up as an outlier, because we did all these different comparisons. So this gene, for example, we find in 2 comparisons significantly outside the range. In all the others, it's not. It's on the range, it's very high maybe, but it's not outside the range. So we think this one, we know this is true. We know this is definitely a gene that changes evolutionary pressure, but it's local. It's not really moving with the species to do anything specific. Whereas this other one, there we go, you see all the arrows all over the place. They all point in this direction. So we think that this is the gene that basically lost its selective pressure or gained new selective pressures here when the species evolved, when there was this migration happening. So this kind of gene is what we like to call our main or habitat adaptation NLRs. And there's another example here. So this one is very clear. Basically on the coast here, we see that there must be something happening because this gene, this allele was always different when we compare the coast with any of the other populations. So now the main question that we have is, how many of these main habitat adaptations are there? And that's actually very few. So here you see the summarized numbers, just because I cannot draw all the arrows. So we actually have only 4 when we move from our center here down to the coast. We have also only 4 from the center to the mountains. We have a few more when we go up north, probably because it's a more complex situation up here. And they're actually overlapping. So in total, there are only 17 of these main habitat adaptation NLRs. And what is very exciting is when we then look at what are these NLRs, and we actually see that they represent different classes than all the other NLRs that are either not under selection or that are local adaptation NLRs. So what we expected at first is that we find that the NRCs, be very conserved, are not main adaptation NLRs, but we actually see the complete opposite. So we see that NRCs are very much overrepresented as main adaptation driving NLRs. And we think now that this actually makes sense because once you move into a new area with a new climate, maybe your whole system needs to readjust. So maybe your NRC also needs to co-evolve with the rest of your network. So we see all these local NLRs belong to many of these sensors, or they're not assigned to plates, and the main NLRs are very often TNLs or these helper NLRs. So this is really exciting, and I think this is the first cool findings that we have with this Solanum chilensis system. So we're very clear proof of sequence evolution. We were not able to test presence absence variants with the data set we have, but we hope to tackle this in the future. We can definitely say that those NLRs that we consistently find throughout the populations adapt locally to biotic stresses due to sequence evolution, but that very few of them really show strong species-wide changes of selection. So only very few are what we are calling major genes, and surprisingly, these hubs, these central NLRs in the network, are overrepresented in the hubs. So also this is an evolutionary relevant finding, because this also means that network position being central in a signaling network doesn't mean that you cannot evolve. And I think that's also a very important take-home message here. Right. So for the last 5 or so minutes, I would like to switch to a different topic, which I started like a year and a half ago as a group leader now. to which a lot of people have contributed: Parvinder, Andrea, Michael, and Lina, and a lot of other undergrad students as well. And it changes focus a bit, because when we're out in Peru or in Chile, we actually see a lot of quantitative resistance in these plants in the field. So what we now want to do is we really want to understand the differences in quantitative resistance between these plants, and maybe also eventually link it to the differences in the pathogens that we find in the field. And I'm only going to show a little bit of a teaser here, because we're still working very hard on this. But we've now perfected, or Parvinder, the PhD student on this project, has now perfected the art of infections by doing several thousands of infections on detached leaves on many and many of these populations. And what we can see is that there is definitely reproducible quantitative differences between our plants from even the same accession number, right? So here we have 10 plants from one location, and we see that there is variation here, but there is always one plant that is always most resistant, and there is always one plant that is always most susceptible. And it doesn't matter, we can also take another population. This one comes from the south, and we see the same. So we always have a most resistant plant and a most susceptible plant. But overall, this whole population has a bit higher susceptibility. It's definitely easier to be infected by Phytophthora infestans in this case. And what we're now trying to figure out is what are the actual mechanisms, so the defense mechanisms that the plants have, that we can link to these kind of differences that we observe. So we're not looking at the RGs that are involved in this case, or the sensors or the receptors or anything. We really look at the very end. So one thing that people often measure, especially in Arabidopsis, to say a plant is resistant, is the production of reactive oxygen species. The more ROS, reactive oxygen species, are made, the more resistant. At least that's the theory. What we find in our plants is that this absolutely does not hold true. So we find here, this is one population again. Here is our most resistant and most susceptible plants. There is a little bit of a difference, but what we find is definitely there is one plant that has, 3 plants that have much higher ROS, and there's even one that is completely impaired, has no ROS burst whatsoever, yet it still doesn't get infected all the time. So there definitely is more. So in the future, and at the moment actually, we're really working on expanding this. So we want to screen more populations, want to screen more responses, other hormones or hormones, metabolites, etc. Want to look at different elicitors to really put everything together. And at the same time, together with population genetics, we are resequencing a lot of these populations to add the genomic information. So if anyone has ideas on how to scale up these kind of screenings, suggestions for large-scale screens and collaborations are definitely welcome, so that in the end we can really model the defense diversity in Solanum chilensis. And before I finish, I want to show that we also have our own pathogen collection experiments. So we also started about 2 years ago. I guess there must have been something in the air at some of the meetings. So we've been to Peru and Chile now 2 years in a row, and we've been sampling a lot of different locations from the southern highlands and coastal areas all the way up to the north of Peru to see what our variation is when we branch out to slightly different but related tomato species. And this is still ongoing work. We're visiting almost 80 sites. We've collected more than 1,500 leaves and are now down to 500 purified samples that, in collaboration with a lot of people in Latin America and Marcotines in Frankfurt, are being analyzed. And with that, I'm at the end. So my conclusions, I already stated them for arginine evolution, and I think in the defense response, by looking systematically at this species, we can really start to learn a lot of new things. And I hope that maybe in the next year, when this series keeps going, I can tell you more about this. So thank you for your attention, and thanks to all the collaborators on this project. So with that, I'm at the end. Thank you.
Thanks a lot, Remco. Yes, applause for this very nice talk. Again, I will pick a couple of questions, and again, if you want to discuss with Remco afterwards, so use his Twitter handle EvoMPMI in combination with Plantae Presents hashtag. So one of the questions is a technical one, and maybe you can combine it with another question from myself. So I was wondering, I mean, you got only 53 genes, which is not much as outliers for the NLRs. And if you would look at other gene families or other genes that are known to have like adaptive responses, for example, to drought or something like this, if you would get to a similar amount. And in combination with this, Matthias Benoit was wondering what kind of control genes you used for the nucleotide diversity analysis that you used on the slide 11.
So the first part of the question I cannot answer because we were very limited in our budget. So we just don't sample enough. But we assume that because we don't find that much. And that answer lies in the second part. So the control genes that we use are a set of genes that people have historically used because they thought they were involved in development. So it's a set of about 50 genes that have been designed in the chosen in the early 90s or late 90s for phylogenetic analysis of tomatoes. But now it turns out that some of them are actually really not so conserved at all because people start to study more and more tomatoes. And it's exactly those genes together with our NLRs that we used to make the demographic model and that we use to simulate. So we basically simulate based on NLRs plus a few extra genes, a range of FSTs, and then we still find stuff that's outside. So I would think that if we now do whole genome scans with the same kind of analysis, that we really wouldn't find that many outlier genes. But I cannot tell. I mean, there is definitely a lot of adaptation happening in these plants, especially to drought and especially to UV radiation, because these are very extreme places. So it might very well be that there is also 24 or so genes that are very important in drought adaptation that these plants cannot go without. I mean, you've seen the pictures, these are extreme climates. So
there are a couple of questions, including one from Pingtao Ding and some others. So they're wondering about hybrid necrosis in tomatoes. So probably they got also a bit motivated by the first talk. So can you give some statements on this?
I don't think anyone looked at it in tomato. Might well be. So I know that when I try to cross plants from my main central population that we're most often using, 3111, with plants from a population 4330, that in a lot of cases we do not get fertile seeds or offspring at all. So or no seeds at all in the fruits. So yeah, there's definitely something there. But we haven't checked it systematically as Detlef has done in Arabidopsis. So I can't really help.
So Remco, there is Leonie Moyle. She has done a lot of crosses, and so from her work, it seems that in tomato you much more often see infertility.
Yeah.
Why infertility is really rare in Arabidopsis is unclear, and in tomato it seems to be the major phenomenon, infertility.
Thank you.
Thanks. Ute Kremer is wondering what kind of pathogens you observed in the field while sampling.
So the most dominant pathogen that we see are Alternaria species. So we see a lot more. I mean, when we start to look closer, so we isolated successfully. We isolated botrytis. We isolated several of the Fusarium species from the leaf or from the stems from the plants because we took some of those. We've seen some Phytophthora, but the most dominant are Alternaria or Stemphylium or anything related.
Feng Du has a question, and maybe it also relates a bit to some of the questions that Detlef got. So here the question is whether the immunity mechanism between tomato leaf and fruit is different.
I don't know.
Again, coming to the tissue and cell type dependent effects.
I don't know actually. I haven't heard much of people actually testing like these typical HR resistance responses in the fruit. I know it exists, but no, I can't answer this.
Alexander Goldsmith is wondering whether within your 14 populations that you collected, whether there's a relationship between level of resistance and fitness and yield.
Wow, these are all very good questions. And the thing is, I've only been me in the lab, so I would like to test all these things, but I just don't have enough hands. I mean, fitness is an extremely complicated thing to measure in this kind of scenarios, because as you might have seen, some populations that grow at 3,500 meters, what kind of common garden experiment do you put them to actually measure fitness, right? So what we know in terms, if we grow them in our glass house, I think this is part of your answer. If we grow them in our glass house, all the plants start flowering after a while, and if we don't cut the flowers off, we will get fruits eventually. And it doesn't really look like one of them gives more fruits than the other, although there is a bit of a delay. So we clearly see that the populations that come from the south flower earlier than the populations that come from the center. But that's about all we know. So we haven't really had time to look into these kind of things. We want to, together with population genetics and with some other people, but these are horrendous experiments to do, and yeah, it just never makes it on the priority list.
Maybe this is a question to, or possibly a question to both of you. So Oliver Arts is wondering whether we can use the vast amount that we have on the Arabidopsis genomes to basically create a pipeline also for other species such as Solanum, and to inform hypotheses specific to some Solanum species.
I don't quite understand.
Well, in principle, whether the genetic information that we have on LRRs in Arabidopsis, for example, if we can use this data also for other species. I think Detlef's talk also kind of touched on this, that there might be some conservation.
Okay. Yeah, I can answer a few things. So we know that in the Solanaceae there's definitely a kind of different way of how these MLRs function, right? This NRC clade, you don't find it in Arabidopsis, for example. So we can, in that sense, we can use some of the findings, and correct me if I'm wrong, Detlef, but some of these findings we can use to phrase new hypotheses and to test how universal they are, right? So the amount of presence-absence variation in Arabidopsis is striking. Does this hold up when you look within one tomato species, for example? I'm not sure, because tomato just evolves differently. The populations are very diverse, so do they need that much copy number variation? I don't know. These kind of things you can test, but I'm not sure if you can take evolutionary models and directly translate them from one to the other. I think mechanistically, in some cases, yes. Like, of course, dimerization, and you need some components to proper function, and you have co-receptors, etc. But in terms of evolution, I think it's more tricky.
Yeah, I think what we would need is some understanding, predicting basically what NLR proteins interact with, and we are still very, very far from that to predict that from first principles.
Correct. Yeah.
Thanks. Arpan Mukherjee comes back to some questions on LR expression regulation, and wonders how day and time and temperature fluctuations might play a role there. So I guess that was motivated by the different settings where you sampled the different populations.
This is one thing that I would really like to test. So I think one of the main reasons why we pick up the NRC as evolving throughout the range is actually adaptation to the climate and not adaptation to something else. So we know that in the south the daytime temperature is higher, definitely higher, and that also the NRC clade itself is the functioning of the NRC clade is temperature dependent. So when it gets too hot, some of the NRC dependent genes do not function anymore. At least the ones that we know. So the ones that have been cloned or bred into tomatoes. So I2 is one of the examples. It's the resistance gene against Fusarium. It works very well up to 26 degrees, 27 degrees, and then gone. And in Chile, it's about 28, 29. So the breeders, they don't use it there. So, but the wild tomatoes, they're not fully attacked by Fusarium. In fact, the wild tomatoes are growing right next to the cultivated ones. The cultivated ones under a tent, and the wild ones next to it in the gutter, and they're still resistant. So there might be something with temperature regulation going on there, which should be testable.
Amira Susana Del Valle She wonders whether you, besides looking at pathogens, whether you also found some endophytes and whether you are looking into this as well.
I have to disappoint there again. So when we went to the field, it was 2 people. It was me and either a PhD student or a collaborator from France, Philippe Riot. And you just don't have time to do this. So it's extremely tedious work to trace down the populations, to make all your notes, to collect the leaves, prepare the leaves so that you can bring them back to a lab, etc. So we didn't look into endophytes. With a very specific sampling regime, we take leaves, we take slices of some parts of the stem to check for Ralstonia, bacterial disease, and for Fusarium, and we take a few flower buds or not. If something interesting there, just to document, and that's it. I'd love to go back with the whole army and check everything that grows on these plants and surrounds it, and make better notes and put it all in an ecological context. But that's for the future.
Yeah, great. Sounds good. Thanks a lot, Remco, for the very nice talk and also for the great discussion here. And well, thank you both that you in the end even joined together with the discussion. I think that was really great.
Thank you.
Yeah, we saw two very nice talks, and they were nicely complementing. And yeah, I was happy that you both joined us. Thank you.
Thank you for giving us the opportunity to present this work.
Yeah. Thank you again. This was really wonderful. And so there were a lot of questions. I tried to answer many of these questions in the chat box. I hope that worked.
Yeah, I saw that. I think that looks really great.
Yeah, I'll be sending you both Excel with the questions, and we can post those answers along with the recording. We are planning to download this and post it to Plantae within the next few days. Thank you all for joining us. This has been really a great experience, and learned so much from this. Be sure to check our website on plantae.org. It's under the Education Plantae Present series to sign up for the next webinars. We've recently added a whole new list of opportunities. So be sure to check that out and be on the lookout for the recording. Thank you all for attending, and we'll see you next time.
Thank you.